In a number of species, particular individuals will lead the group while travelling, providing access to a number of foraging resources. Tursiops truncatus, or the bottlenose dolphin, is one species that do this. To test if leaders in groups may guide relatives, Lewis and her colleagues studied a group of bottlenose dolphins in the lower Florida Keys, as can be seen in Figure 1, whose behavioural traits showed leadership qualities. In the lower Florida Keys, bottlenose dolphins live in dynamic fission-fusion groups, meaning that smaller subgroups can split off to travel, but then these groups can then rejoin or fuse back together in certain times of need. The nature of prey resources in the Keys is ephemeral. There are only patchy schools of fish to be found and restricted ranges. Therefore, the bottlenose dolphins do not socially forage, but instead travel together and forage individually on single fish that are located. Leaders in these groups, therefore, do not benefit from location of prey sources first, or by help of other group members securing the prey. Consequently, it has been documented that consistent leaders guide groups to areas with greater prey availability compared to individuals that are not consistent leaders. To investigate this hypothesis, the potential benefit of leading in fission-fusion groups relate within leader-follower pairs in the lower Florida Keys, a dolphin population was studied. Lewis and her colleagues tracked an individual dolphin group over several years. A video camera attached to an airship, as can be seen in Figure 2, was used to record the individuals in the group and the direction changes initiated by certain group leaders. Collecting data on individual dolphins was undertaken in the study area by monitoring individuals with easily distinguishable fin markings. Skin samples were collected from 36 dolphins during 2008 in the lower Florida Keys study area, as can be seen in Figure 1. The examination of the haplotypes was completed to determine the possibility of a genetic link to individual differences in foraging strategies. The researchers wanted to determine if particular dolphins led the group more often than expected, and if the dolphins in the front of the pod controlled these direction changes. Using photo identification, it was determined that individual leaders could be identified in dolphin groups by their presence in the front of the growth, both during surface events and subsurface travel. Samples of genetic material were collected from the group of dolphins in order to assess mitochondrial haplotypes. Results concluded that 3 out of 4 dolphins possessed the same haplotypes. This finding supports the possibility of a genetic link to individual differences in foraging strategies, and certainly is consistent with the notion that genetically determined personality traits maintain a role in the acquisition of at least some foraging strategies.